Hello students, today ERQ is evaluate research on the role of neurotransmission in human behavior. So as you know that this ERQ is from biological approach and the command term is evaluate. So that means we have to write the strengths and the weaknesses of the role of neurotransmission. So first we will start with introduction that carries 2 marks, and this is what I write in my ERQ. That is the introduction, and I write this in my paragraph. Neurotransmission is a process of communication within the brain. Neurons release chemicals known as neurotransmitters in response to stimuli or internal changes, which are then transferred to electric signals. Different neurotransmissions are known for different effects in the brain. For example, acetylcholine plays a role in the consolidation of memory. Roger and Kessner and Etanova et al. Both studied the role of acetylcholine in the consolidation of memory and together provide solid evidence that acetylcholine plays a role in consolidation of memory. So I write the first supporting study after my introduction and aim of the ERQ. Roger and Kessner's study aimed to determine the role of acetylcholine in the formation of spatial memory. There is a high concentration of acetylcholine receptors found in the hippocampus, an area of the brain responsible for the formation of memories. The study looked at the formation of spatial memory in 30 rats. The rats were randomly allocated into 2 groups. The treatment group was injected with scopolamine, which blocks acetylcholine receptor sites. The control group was injected with a placebo saline solution, as there was a possibility that the injection itself changed the rat's behavior through adrenaline or stress. They were then put into a maze. The results showed that the treatment group made more mistakes, which suggested they took longer to learn the maze. The next is the evaluation, as the command term says. So I evaluate the supporting study, and this is what I write. The study manipulated an independent variable and tried to control extraneous variables. Thus, it can determine a cause and effect relationship. It is highly standardized and has high reliability. However, due to the simplicity of the study, there is no triangulation, lowering credibility. The lack of theory triangulation also highlights the potential weakness of the study, reductionism and ecological validity. Because the study's goal was to ultimately relate to the role of acetylcholine in human behavior, the choice of rats provides a problem with ecological validity. Whether this study can be able to be generalized to human behavior is unclear, as cognitive processes such as memory may be more complex in humans. The next is the second supporting study that I write, that is by Antonova et al. Supported Roger and Kessner's study on the role of acetylcholine. They wanted to research the effect of acetylcholine on encoding spatial memories in humans. The study used a sample of male adults. The participants were randomly allocated into 2 groups. The treatment group was injected with scopolamine and the group control injected with a placebo. The participants then play a virtual arena game, and brain activity was scanned with an fMRI during the activity. The activity consisted of participants finding their way to a pole, and then with a wait of 30 seconds, the participants would be set in a different location and find the pole again. There is a question of whether this activity is ecological valid, as this activity is quite artificial and may not represent spatial encoding in real life. The participants returned 3 to 4 weeks later and repeated the procedure, but received the opposite treatment. By using repeated measures, Antonova eliminates participant variability. The results showed that participants had lower brain activity in the hippocampus when injected with the scopolamine solution. There was no significant difference in the rate of error between the 2 groups, although scopolamine did have more errors. This suggests that although the fMRI provides strong biological evidence, the actual arena task itself may not properly reflect spatial memories. The study was counterbalanced, meaning that participants did the 2 treatments in different orders, eliminating the potential and the practice effect. The study was also carried out blindly, eliminating researcher bias. Research provides empirical support that acetylcholine plays a role in the consolidation of memory. Now, this is the evaluation part, that is an important one that you have to write in this ERQ. So it goes like, research provides empirical support that acetylcholine plays a role in the consolidation of memory. The research has high internal validity. However, cognitive processes such as consolidation of memory are abstract concepts, and models can often be reductionist in terms of explaining how memory works. Thus, due to the complex nature of memory, research lacks ecological validity, as experiments have to attempt to limit as many potential confounding variables as possible. Research on memory also may have difficulties with construct validity. For example, in Antonova's study, although there was a significant difference in levels of brain activity in the hippocampus between treatments, there was no significant difference in the actual amount of errors, which is what would actually be significant in the real world. And last we write is the conclusion. In conclusion, research on the role of neurotransmission in behavior, specifically acetylcholine on consolidation of memory, is quite strong and has high internal validity and reliable results. Whether these results can directly be generalized to humans was a concern in Rogers and Kessner's study, but Antonova's study minimized those concerns as he found biological support for the theory. Research on the role of neurotransmission does have potential concerns of ecological validity due to the difficulty of isolating and operationalizing variables in the complex abstract process of memory. I hope this ERQ helped you, and if you have any queries, please feel free to write your comments in the comment section below.